All right, we're back. I'm going to finish up part 5, and then we will get to the cross-reactivity study that I had. I just did a quick last-minute rearrangement of these things. So the thing is, if you look at the way T cells work, they work on snippets 10 to 15 residues long. This has a plus side and a downside. The antibodies work on intact proteins outside the cell. T cells work on cut-up proteins from inside the cell, in the context of MHC molecules. If you think about this, and we talked about this a little bit with the cancer example, a few mutations might get rid of a couple of T cell peptides, but there will be other peptides that are unmutated. As long as you have a 10 to 15 residue stretch that binds an MHC, that's all you need at the minimum for T cell immunity to take place. And there's a lot of 15 residue stretches that can be recognized by the immune system. So this is why my reaction was when we saw those things about antibodies waning and you can have mutations that will destroy antibody responses. All true. But the fact of the matter is the T cell response is much harder to completely avoid. And in fact, there's even there was evidence at the time that there are some of the T cell epitopes from common cold coronaviruses that are the exact same as SARS-CoV-2 epitopes. Because if you have 60% identity, then you're going to have some of those T cell snippets are going to be the same. This is the way to think about it, and this is thinking about immunity like a biochemist. That's what I want from you. So this is why I was, researchers are looking beyond antibodies for clues to lasting protection. Yeah, we've got to look beyond antibodies. And again, the T cells are slower. They won't prevent you from being infected, but they should, at a level, prevent severity from kicking in. This is the exact one of the references, and so this was one of the things. This is more than a year old now. But people who have been infected with SARS-CoV-2 are generally going to have T cells against 15 different peptides from the coronavirus proteins. That means if one of those gets blown up by a mutation, you've still got 14. If two of those get blown up, you've still got 13. So it's like you're taking a hit, but you have margin here. And that's the important thing. It's not as bad as it sounds, especially if you're just looking at T cells. This is that paper that was being referred to, so if you want to look at exactly the data that comes from that says that most people respond to at least 15 peptides from this one virus, this is where it comes from. This is Alessandro Sette's work, and you can actually find even seminars about this online. But here's the idea, and the one main thing is notice here you have CD4 cells and CD8 cells. CD8 cells are the cytotoxic MHC class 1 responsive cells. CD4 are the MHC class 2 helper T cells that you're talking about. And they actually look at that, and you can actually see the data. So the big picture, we recently have I don't have the date of this, but this is like a month old at most right now. And this is the latest study of not how do antibodies work, but how do T cells work and how do T cells remember. And it compares being infected with COVID before having the vaccine versus having that reversed, and how does that affect the T cell response. And there's one surprising result, which is optimistically surprising from this. The scientist who did this, Thomas, has a whole Twitter thread about it, and this is what Twitter is good for. It can get me mad about the things that people say that are wrong, but then it has scientists that are doing little explainers, which are nice. And what they came up with this is, notice how they measured what the sequences of TCRs are that respond to COVID. And they've got over 4,000 sequences of different TCRs from different people. This gives a pretty good view of how does being infected or vaccinated against SARS-CoV-2, how does it affect what T cells you have to employ against the virus. So here's one of the main things that they have here. Pink means you have more of a response. Blue means you have less of a response for the average expression. And the thing I want you to notice about this is the T cells that respond to the spike proteins versus the non-spike proteins, everything else. And at the bottom you have all sorts of different cases. Infection alone, infection before vax, vax alone, vax before infection. And if you look at that, you can see some really interesting patterns. But the main thing that I want you to see is look at the last 4 columns under spike. Okay? You see a lot of pink. And you see more pink here than you see under non-spike. And so you see that you actually, if you continue to get either infected or vaccinated, you're going to get these molecular markers that, number one, you have cytotoxic T cells, and number two, you have memory cells that are remembering what the spike proteins look like. What's also cool is if you look on the other side, you see a little less pink because non-spike proteins are not included in the vaccine. But even though they're not included in the vaccine and they're here less, you still see some pink. You still see some responses to it. And that's what the nice surprise is to this. We're getting more responses to non-spike proteins than I would have expected at minimum. So we are getting, basically the response of the T cells is broader. It's not just against spike. It's also getting some pink against non-spike, even if you were only vaxxed twice and then infected or things like that. So, and you can see there are different T cell profiles. We're not going to get into the different types of T cell profiles, but you see there are some patterns here in which you have some of the pinks are on the top. That's one type of T cell. Some of the pinks are on the bottom. That's another type of T cell. Immunology, it's great. But here's the really interesting thing. So like, what if you're infected and then you're vaccinated? What happens to your T cells that you get? And you see right here that you have, after you're infected, most of your responses are to the non-spike proteins. Then you get vaccinated and you get a bunch of responses to the spike proteins. The pink part on the left grows in. But the infected, but you see that you still have a significant amount of non-spike proteins. The graph on the right is what is a really interesting one to me. It shows the situation, the same situation is shown on the left as on the right for the pink and blue, where you have an infected response, where you have a proportion of spike-specific T cells. You have a lot of different things for a lot of patients. Some people make a lot of spike-responsive cells. Some people make very few spike-responsive cells. You see that it goes all the way up and down for the pink. Then if you're vaxxed, you get, in that context, you get a dose of the spike protein. Then all of a sudden, you get a bunch of T cells that respond to the spike protein. And so you get the blue thing. So if you're infected then vaxxed, you get a bunch of responses. So you see at the top of the blue part on the right, you have this big bulge where you have a lot of spike-specific T cells. It's like your T cells were already on alert. Then you gave them more spike, and they're like, oh, I'm going to look for this guy double time. What's really interesting to me, though, is the yellow. The yellow is for the case of somebody who was vaxxed twice and then had a breakthrough infection. The one caveat is this is with Delta, or Alpha maybe. This is not with Omicron. But from what I've seen, I expect Omicron to work the same. What's interesting about this is that you have a lot of responsive T cells, and, at least 10% of them are responsive to spike. But what's really interesting about this is the system, the body, saw spike first, and then saw everything when it was infected. You might expect from that it would keep on responding to spike. But instead of being like the infected then vaxxed version where you have all these spike responsive ones, you actually have a broadening of the response where you have a bunch of like 10% spike responsive T cells. Even the bottom point on the yellow dot is down around 10%. So that means that you have 10% spike specific. But that means 90% against the other stuff, which is the stuff that really gives you a broadened protection. You're not just protecting yourself against spike. And so I wasn't sure how this would turn out. I wasn't sure if the yellow and the blue on the right side would be the same or different. But it turns out that what this is actually pretty positive, because it means even if your body sees spike first, it's smart enough to broaden the response to non-spike and produce a lot of non-spike responsive T cells that will protect you more widely than just T cells against spike will do. This also means that if you have a mutation in spike, but you have a T cell against non-spike, guess what? It's going to be able to still protect you against that. So this is what the good news is. The good news is that the yellow line is high, but it's bulged at the bottom, not at the top. Good news. There's the bulge, okay? The other thing that they did, remember, they got the sequences of each of these T cells. And so they were able to see, do people have recurring patterns, recurring sequences that keep showing up in their T cell receptors? that show up over and over again against the same antigen, the same virus that they're responding to. And they found out, yes, they do. You see, they have these clusters of T cells in different people, and they all have this same kind of sequence. And you see it's like a logos representation that, like I showed in Biochem 1. And you can see that they're not exactly the same, but they're enough the same that you can see, like, you can see that you get this sequence SL something GGTEA. And you get that in a lot of different people. So the same T cell sequence, which is in the loop region, which means it's recognizing the same kind of pattern. Now, these are for people who have the same MHCs, and they're able to correlate the T cell with the MHC allele that they have. But the thing that I want to show you here is look at all the colors that are on the right side. The colors tell you whether the person had been vaxxed first and infected, or if it's green, they'd only been vaxxed twice and never infected, and if it's pink, they were just infected, things like that. The main thing I want to show you is that all these same sequences are showing up in people with all 5 colors. You see all these different colors on the right side. That means that people, no matter whether they were vaxxed or infected or neither or both or they're producing the same sequences of T cells, at least for these particular cases. This means that we have the hope of analyzing your T cells, sequencing your T cell receptors, and saying, oh, you've got this sequence. That means that you have a T cell that will protect you against COVID-19. And it won't matter. Some of them might be different if you're vaxxed or if you're infected. But the main thing is you have a lot of different colors. So whether you're vaxxed or infected, you're making a lot of T cells. And we're going to figure out what sequences of T cells go with what kind of protection. So the scientists had this, and like nobody cares. But when I see something like this on Twitter, I'm like, oh, I've got to check this out because it's for just you lovers of T cell receptors. Okay, that's me. Guilty as charged. And basically, this Dr. Thomas is talking about how many unique TCRs they have sequenced, covering 18 different epitopes from the virus, so that we can sequence a TCR and say, hey, that TCR recognizes this specific epitope in this specific MHC. So this means we can have diagnostics that will look at T cell receptors. It's just harder, and it takes a lot more work. But with Dr. Thomas's work and others, we're getting there. There's actually a company that's working on this. They're a company that's local. I even know someone who works for them, Adaptive Biotechnologies, and they have a T-Detect test. Now, it's more expensive than an antibody test because it's more complicated. But they take your blood, they sequence your T cells, and they look to see what kind of sequences are in your T cells if they are associated with protection. And this is all the way back in December 2020 when they first rolled this out. I believe this has been published now. And this is at least what it was back then. I actually haven't checked it recently, but you can test to get your T cells analyzed. And I sort of want to pay up for this. I'm pretty cheap when it comes to these things, but I'm not sure if I can resist this, so I'm going to think about that. The conclusions is that remember how T cell immunity works. It works on peptides, not on intact proteins. And so we can actually build these in the lab. We can look at and do experiments to find out where immunity is located and what sequences of what receptors provide immunity. This will allow us tracing the course of infection and hopefully someday understanding why some people are more at risk than others, so that we can give people an accurate view. I seem to think there are some people that say, oh, I'm not at risk at all. And there are some people that say, I'm so at risk I'm not going to ever leave my house again. Between that is where most of us are. Some of us are actually at one of those ends maybe, but most of us are more in the middle. Where are we? We can find an answer if we do things like figure out how our T cells work. So that is the part 5.